So before we get started, I wanted to do a quick shout out to some of my colleagues in a paper which they just published, Microbes Scared to Death by Virus Presence. And this is my colleague, friend, ex-Portland resident, Rachel Whitaker, who's at the University of Illinois Champaign-Urbana. And they're working with viruses that are near and dear to my heart because these are the lemon-shaped viruses that we work with mostly in our lab and that I originally discovered in Kamchatka and Tien's actually working with right now. Unfortunately, I disagree with their interpretation of these results, but that's a completely different story that we can talk about some other time. And actually, we know almost nothing about virus entry for these viruses, which I think would be a really interesting question, and maybe that's how they're scared about these other viruses which are there. And so these are these particular viruses which we have over here on this side. So of course, the reason everybody's here is our clicker questions. So let's go ahead and move to those. Actually, it would help if I turned on the clicker. That might help. We'll see. So it gives you another second or two to get your clickers out. I did finally get the feedback from molecular biology from last term, and the jury was out on whether to do online quizzes or clickers throughout the class, etc. So, of course, lacking consensus leaves it kind of up in the air about what we actually want to do. So today's is viruses with the biggest genomes known to date are dsDNA viruses, ssDNA viruses, SS-positive strand RNA viruses, SS-negative strand RNA viruses, or retroviruses. And again, feel free to talk amongst your friends, colleagues, enemies, etc. Ten, 5, okay. So by far and away, the largest genomes are present in double-stranded DNA viruses. Yes. One of the comments that I got from last term was that the clicker questions were completely unfair because if somebody had their laptop and the notes from previous time, all they had to do was Command F and try and find the keyword from the last lecture. I don't think biggest was in the keywords for last time. Actually, I don't think it's anywhere in the lecture. So hopefully that didn't work this time. But still, all of you got that, which means I'm a fabulous lecturer because I got it all to you, the appropriate information. Single-stranded DNA viruses we'll talk about on Friday, which is a reminder that next week from today and a week from Friday will be guest lectures / videos. There probably won't be clicker questions for those days, but there also won't be YouTube recordings for those days. So just a warning as far as that's concerned. So these single-stranded DNA viruses are really fascinating, and actually my lab has gotten really excited about working with these things. And this is, again, kind of an aside here. Some of the other comments, Stedman has a tendency to go down rabbit holes. This is one of those really quick rabbit holes. But it's, I think, a great reason why you should have faculty members teaching classes and doing research at the same time, because the only reason that we were really able to jump on this new project and this new virus that George will talk to you about next week, because I was teaching about single-stranded DNA viruses in this class. I had no clue about single-stranded DNA viruses before I started to teach this. And so that really tied into the research, I think, extremely well. So it's not just the research helps the teaching. I think the teaching also helps the research. So again, little shout out for why, when you're taking classes, you should demand that your professors are actually doing research as well as they're doing their teaching. So yes, our next question is, most common cellular mechanism used for animal virion entry into host cells is phagocytosis, pinocytosis, endocytosis, hemagglutination, or pore formation. And again, feel free to discuss this with your neighbors. With the green end or the other end? Green end up. Got it. Yep. So 10, 5, so why is it not phagocytosis? I heard some mutterings. Why is it not phagocytosis? Because phagocytosis is usually for big stuff, much bigger than most virions. There clearly are some virions, like the ones that have the absolutely most massive genomes, like the double-stranded DNA viruses, the Mimi viruses, et cetera, which almost have to come in by phagocytosis. But otherwise, as most of you have correct, and again, because I'm such a fabulous lecturer, endocytosis is the main mechanism whereby viruses get inside the cell. And again, it's a completely cellular process. Pinocytosis would be used for what? Tiny viruses. And there are a few tiny viruses for which this is the case. Hemagglutination. So that's a viral mechanism. It's not a cellular mechanism. And it is certainly receptor binding. So it's receptor binding by the viral receptor binding protein, in this case sialic acid. Pore formation for mostly bacterial viruses. There is, however, one animal virus that does this. Polio, exactly. So polio and a number of the pico-RNA viruses which do this. So those of you who have to leave, adios. And I know people were, oops, hang on. Let's make sure we get the right one here so you actually get the right points for it. Good. So this is basically, again, a review of what we had last time. Envelopes, again, these are what's present on the outside of your virions, have integrated membrane proteins in them. These are going to be your receptor binding proteins. And we'll look at those in a lot more detail today when we talk about some of those entry aspects. Assembly versus disassembly, again, this is an absolutely specific viral process. It's how you put the capsid together and how it remains stable on the outside of the cell and how it then gets degraded on the inside of the cell. So you have to have this metastable state for any of the virion particles. Completely separately was virus taxonomy. Ugh. This is the book, actually this is the 8th report of the International Committee on the Taxonomy of Viruses. There's now a 9th report, excuse me, which is available that you can borrow from my office if you really want to figure out a lot more about any particular kind of virus. But again, these are excuse me, mostly split up. You may be able to see in the back, or not in the back, more in the front, different color coding on the back. These are the various different single-stranded RNA viruses, single-stranded DNA viruses, double-stranded DNA viruses. This big one right here is the single-stranded RNA viruses, which are in there. So mostly, at least in terms of virus taxonomy, people are splitting this up based on those genomes. Do they have envelopes? Do they not have envelopes, et cetera? What I think is probably a much better way of thinking about overall virus taxonomy, where they originally came from phylogenetically, has to do with these structural classes. So we talked about the STIV and the capsid protein, which is basically identical between all kinds of different domains of life, but that has no sequence similarity. Nobody classes these viruses together, at least not the International Committee on Taxonomy of Viruses. So that, I think, is probably a really interesting area that we're going to hopefully be moving into in the next couple of years, thinking about structure in terms of how things are related to each other, which is kind of funny because that's what the macrobiologists always used to use for classification purposes as well. And so now the nanobiologists are moving to thinking about classification, again, based on shape, based on physical characters. So it's an interesting process. In terms of entry, we talked about receptors and fusion, and we'll look at that in a lot more detail now through the rest of today. We didn't have much of a chance to get into intracellular transport. We'll be doing that a little bit more today. So again, big picture in terms of virus entry. It's all about virus receptors, which are cellular proteins or other macromolecules. Classic example, sialic acid. It's not a protein, it's a sugar that the virus receptor bindings proteins bind to. Membrane fusion, which is really critical in terms of enveloped viruses. Once you get to the inside, getting rid of the coat is a major issue that all these viruses have to deal with. And then very often, at least in eukaryotic cells, you have to get this genome to a particular place. So we'll look at that. And then towards the end, we'll look at some antiviral drugs, which are very specifically directed against a lot of these processes in terms of how these things come inside the cell. So I updated Flash on here, so now we should actually be able to look at some of these animations. Here is the animation of an enveloped virus binding to receptors on the outside of the cell with its receptor binding proteins and undergoing fusion, releasing this capsid here into the cytoplasm. Of course, you still have to get the genome out of this nucleocapsid in order for that genome to continue to replicate. This is one process which is used. This is a slightly different modification of that process, which is where you now go into an endosome inside the cell, and then you have membrane fusion that happens once you're inside this vesicle, and again you have release of the virion at that process. And so this is all again about membrane fusion taking place as it's going through here. You also have receptor-mediated endocytosis, for these non-enveloped viruses. These then also somehow have to get out of the endosome, which of course is magically dissolved in this particular case. And we'll look at these examples as we move along for each of these individual viruses, how they go about doing this. And so that's the very gross cartoon version of how this works in terms of real data on how these receptor binding proteins are interacting with the receptors and causing membrane fusion or uncoating of the genome is best understood for influenza. And these are the influenza receptor binding proteins right here, known as HA. HA stands for what? What have we talked about in terms of assays so simple that even I can do them? Hemagglutination. Hemagglutination, HA. So this is the protein which is important for hemagglutination. And anytime you see like H5N1, for instance, talking about different kinds of influenza viruses, H is the HA. So it's the fifth kind of HA. So this HA starts out as HA0 to make it nice and complicated, then becomes HA1 and HA2. This is what's involved. I mentioned really briefly maturation. This gives the option to come back and talk about it again. This is a protein which is produced in one form but becomes a second form in order to make the virion infectious. How it goes from 1 to 2 is proteolysis. And again, we'll be hearing proteolysis again and again and again and again and again throughout the rest of this course. So proteolysis takes this one peptide, chops it into 2. The way that this protein works, this blue part at the end, is what binds to the receptor on the outside of the cell, which is what? Sialic acid, and what pulls those blood cells together, but also it's how the virus is recognizing cells that it can replicate in. And then, under low pH conditions, which is what you get in the endosome, so now the virus knows that it's inside an endosome as opposed to on the outside of the cell, then there's a conformational change that happens in the protein. And conformational change is also very, very common in these receptor binding proteins. And that conformational change basically takes these short alpha helices here, extends them, and it's a really massive conformational change that pulls the membranes together. Turns out that this is the case for influenza, but it's also really well known for HIV and for dengue. So we'll take a quick look at the HIV cycle right here. This is, again, flash should now be working again. This is from the Harrison lab, where we're now looking at these interactions between the two here. Now, this is the dengue one. Where's the entry of virus into hosts? Maybe I don't have it here. But dengue fusion is another fusion protein. Functions a slightly different way in terms of getting the membrane fusions to come together, but it's also really quite well known. So here's your dengue virus particle, interacts with receptors on the outside of the cell, gets incorporated into the cell, and then this vesicle is transporting this towards the nucleus. There's a change in pH. That's what these little starbursts are supposed to be. You see a big conformational change in the proteins on the surface of the virion. These are now competent for getting membrane fusion to take place. We've got these hydrophobic ends which insert into the membrane now of the endosome on the outside. Then you have these massive conformational changes that take place in these proteins, basically pulling these two membranes together. This is now the cellular membrane and the envelope of the virus. Once you have these two that have bound to each other, now you can have release of the genome, which is on the inside, and that's what these guys are all supposed to be. These genome pieces which are all coming out here. There's not multiple proteins bound to this genome. This is a bit of artistic license here. But that's the process where you have membrane fusion in dengue. Then for HIV, which is probably this, is it the second one here? This should be the proper link. Not quite sure why it's not giving us the right one here. This is still dengue. Why is it not giving us the HIV one? Had it pulled up here earlier. Well, be that as it may, I'll make sure we get the correct link eventually for HIV. But basically all it shows is exactly the same kind of conformational change, only with an individual protein as it's undergoing this conformational change. And one of the things which has been developed quite recently as an anti-HIV drug is a small chemical which fits into that structure and prevents this conformational change from taking place. So it's a structure-based anti-HIV drug which is involved in this infection process. Yeah?
That pH change that's going on in the endosome, is that happening, is the virus inducing that, or is that happening?
So the question is, what's the pH changes happening in the endosome? These pH changes happen in the endosome in all endosomes once they come inside the cell. There are some, not so many viruses, but definitely some bacteria which then get into endosomes. Those will actually modify the pH to some extent. But viruses don't seem to be involved in modifying that pH. Yeah?
So, just to be clear,
Yeah.
the hemagglutination HA0 becomes HA1 and HA2 by being cleaved, and that is the receptor, that binds the receptor on the external part of the cell and becomes the membrane fusion protein?
Yes, so you start out with HA0. That's what the virus produces when you're making the virion in the first place. That then gets cleaved proteolytically into HA1 and HA2. And one of the key things about this is that the maturation process, it's really important that you don't try and reinfect the cell that you've just come out of. And so maturation is really important so that you're going and finding a different cell to interact with. Because if you try and reinfect the same cell that you were in the first place, that's really kind of counterproductive. So that maturation, cutting HA0 into HA1 and HA2, happens very late in the process, basically as you're on your way out making the virion. Then what happens is HA1 and HA2 are still together through non-covalent interactions. You have interaction of the HA1 with the sialic acid, which then causes a conformational change which pulls the fusion protein, which is HA2, together to pull the two membranes together. And we'll see that in a cartoon form. I think, do we have it last time or this time? I forget which lecture it's in. But there's sort of the two blobs that you have there, and they move very much out of the way. We'll talk more about this when we talk about influenza again later on in the term. But you're exactly right. Your summary was exactly correct. Okay, so we already talked about now how you're getting the genome out. We saw the case for dengue in that animation. Have membrane fusion, if it's an enveloped virus and your genomes are just on the inside of the envelope, they're released automatically by membrane fusion. And so that's really pretty straightforward. We also saw transport along microtubules. This is what's happening again with that dengue virus once it comes inside the cell. Almost always you're going to have some kind of replication in the nucleus. These virus particles have to get to the nucleus, or some kind of vesicle inside the cell. And we'll see why that is when we talk about these individual viruses a little bit later on. Occasionally you'll have replication in the cytoplasm, but still you've got to get your genome out into the cytoplasm. And how you do that depends again on the virus. So uncoating is really all just about releasing the nucleoprotein. Again, sometimes it'll be at the cell membrane, you're fusing immediately. Again, that's pretty rare. Very often it's going to be through the endosome, and the endosome is great because cellularly, normally the endosome is undergoing this pH change. And so the virus has obviously evolved to take advantage of this and say, oh, I now know that I'm inside the cell rather than outside the cell. So now it's time to uncoat, as opposed to when you're outside the cell and a neutral pH, we're going to be in the stable state. Pretty much all, with the exception of things like HIV, release their nucleoprotein before they get to the nuclear membrane. And the reason for that is nuclear pores are relatively small as far as virion size is concerned. So if you bring your whole virion all the way to the nucleus, you can't get that virion through the nuclear pore complex. The way that HIV ends up dealing with this is waits actually for mitosis to take place. And then, of course, there is no nuclear membrane anymore. Why bother getting into the nucleus? Because that's where genome replication happens, cellular genome replication. If you're a DNA virus, obviously that's where all the DNA polymerases are, all the precursors for your DNA polymerization. Also, RNA polymerase. RNA polymerase is all in the nucleus. So if you're using any kind of DNA virus, you have to get to the nucleus. And even if you're an RNA virus, which is, in theory, only needs what you have in the cytoplasm, it still needs ribonucleic acids. And those are all present in the nucleus as well. So again, how do you get there? I mentioned before, wait for mitosis. Again, only happens in some of the retroviruses. very often you have nuclear localization signals that are present on particular viral proteins. And in fact, the first ever nuclear localization signals were found in viral proteins and then much later shown in cellular proteins. And often you'll see the capsid gets to the nuclear pore and gets disassembled. All of this is basically present in this figure, which I presented last time, with all of these different models. Here, nuclear localization signals, nuclear localization signals. Here, disassembly at the membrane, disassembly at the membrane. Very small virions can actually get in, these two cases here, and this one down here right at the bottom. Bacteria. No nucleus. Okay, but they've got this really tough cell layer on the outside. Gram-positives have one membrane and a thick cell wall. Gram-negatives, two membranes. Once inside, once you've released your nucleic acid, it's ready to be replicated because replication is happening pretty much everywhere inside the bacteria. So now this one should work, the T4 entry video. And my apologies to those of you who've seen this before, but it's just so cool we have to watch it again. And this is a video of, or animation I should say, of what happens with bacteriophage T4 when it's infecting poor unsuspecting E. coli. So we'll go to full screen here. Here's the bacteriophage T4. One of the reasons that I particularly like this is that this is all based on really high-resolution structural data, both from cryo-electron microscopy, but also from X-ray crystallography. And for some reason, it's not doing full screen there. So we'll just do here. So here's our bacteriophage T4. Here's the poor unsuspecting E. coli's. Comes down, the tail fibers interact with receptors on the outside of the cell. You'll see that here in just a second. Here are those virus receptors. Then you have a big conformational change in these tail fibers, which then attach. Then you have another conformational change, which is the compaction of the syringe-like tail. That brings an enzyme inside the cell, which breaks down the membrane, and the virus genome is released. When I first saw this video, which literally was a few years ago at a meeting in Florida, one of the guys who was involved in it said, "Oh, they totally screwed up." And that is that the compaction that they have there is actually the wrong way around. It compacts counterclockwise rather than clockwise. And apparently they told the animators, and the animators said, "No way, we're not changing it." So, yeah, artistic license, I guess, is what we'll call it. So once it's inside, again, because bacteria have all of their replication, transcription, translation machinery right there underneath the membrane, that genome can be replicated immediately. So we'll talk briefly about some of the drugs that have been designed for looking at this. Basically, two kinds of classes, either blocking binding of the receptor to the virion or inhibiting uncoating. Binding, the classic case here, are vaccines. What do vaccines do? You produce antibodies. The antibodies are going to bind to the outside of the virion, which is going to block receptor-virion interactions. You're never going to have that. Number of people have tried, particularly in the case of HIV, to make a whole bunch of soluble receptors. So basically swamp the system with what the virion will be interacting with. So it'll interact with something on the outside of the cell and never be able to get in. In theory, this works great. In practice, it has yet to work properly. On the other hand, uncoating inhibitors seem to work a lot better. So the so-called channel inhibitors, we'll take a look at this in influenza. This is an ion channel that not just the endosome becomes acidified, but the internal part of the virion becomes acidified, and that's because of this ion channel. And that causes a conformational change on the inside of the virion as well, the nucleocapsid protein, which is absolutely required. If you block that acidification on the inside of the virion, you don't have infection. And so some of the best influenza drugs actually work with this. Amantadine is one of those examples. Few other ones are inhibiting uncoating of both rhinovirus and picoRNA viruses. Here's an example of one of these antiviral drugs binding right in the middle of this channel. Normally this channel would allow acidification of the inside of the virion. Here's this drug that fits right in the middle of it. Here's poliovirus. This is just one of the capsids, the trimeric capsid proteins on the outside. Right here is where a drug was designed such that there can't be that conformational change that you need to make the pore structure in poliovirus. Blocks that, seems to work really quite well. You can also block cellular processes. You mentioned before, endosomes become acidified. You can block endosome acidification. The problem with blocking endosome acidification is, of course, it's a cellular process. So if you're blocking endosome acidification and you need endosome acidification for uptaking nutrients, etc., this can be a pretty major problem. Also are blockers of other enzymes which are coming inside the cell with the virion. Tamiflu got a lot of press when we had all of the scares about bird flu. This actually blocks one of the other viral envelope proteins and blocks its activity. This is one of the interesting drugs that was developed based on knowing the high-resolution structure of the protein on the outside. And so this is sort of modern drug design. You have the structure of one particular protein. You want to block its activity. You then figure out what the structure is of the active site of the enzyme, and then try and design small molecules that will fit in there. And that's exactly what oseltamivir, also known as Tamiflu, does. Binds to the active site on these neuraminases. So these are some of the drugs that have been designed to specifically block this virus entry process. But of course, you have to be careful. Blocking virus entry may also be blocking cellular entry, because they're all using cellular entry processes. Once the virion's been made, of course, it needs to get out. We've already talked about sort of the two mechanisms here. Either lysis, build up all the virions inside the cell to a certain point, and then poof, blow open the outside of your cell, release all of the virions. This is true for almost all non-enveloped viruses, because how do you get across a membrane without bursting open the cell if you're just protein? So cells don't usually release just proteins. Very often there'll be a protein which is important for lysis that's being expressed in the late genes. So as you're making your virus particle, your virions, you also will be turning on a protein which is important for lysis. And we'll see that for the small RNA phage towards the end of today. Of course, if you're making a lysis protein, you've got to make sure that that gets made only after you've made and assembled all your virions, because if that gets made first, the cell is lysed, it's no longer going to be providing all the material you need to make virions. So the timing for this process is absolutely critical. Budding, on the other hand, is something that cells do all the time. They'll release membrane blebs, vesicles, etc. Envelope viruses do this pretty much all the time. How you can release envelope viruses just by budding through the plasma membrane. Of course, timing is not as critical here, because your budding process is just going to be releasing that virus particle. And this ends up happening by assembling, and we talked about this last time, basically the lipid rafts, some particular areas of the membrane where you'll get your virus envelope proteins concentrated, then get either the nucleocapsid or the genome to that particular location, and then bud through the membrane, picking up these virus envelope processes on the outside. So any questions about entry, release, etc. at this point before we move on and talk about cool viruses that infect bacteria? Okay, so RNA phages. This is the book that you could have read instead of the chapter in our textbook. They used to be very well studied. This particular book is in 1975, but if you looked at the work that's been done on RNA phages since then, this book would be almost exactly the same size. So people stopped working on them in the 80s because, who cares about RNA viruses that infect bacteria? Other than some of the applications we'll talk about right down here at the end. So a little bit more about bacteriophage. We talked about bacteriophage before, and again, there's this whole meeting, the birthday party that I was at in January, where all the talks are available online on YouTube. We'll talk a little bit more about the discovery of these single-stranded RNA phage, a little bit about entry, but mostly about replication of these, because that's the really fascinating and different part about these RNA viruses. Our big picture, again, what's a bacteriophage? These particular ones are single-stranded RNA phage, and the big problem, which is true for these RNA phage, but it's true for all RNA viruses, is how to do transcription versus translation, or how to replicate your genome at the same time as you're trying to translate it. Because positive strand RNA viruses, you're translating and trying to replicate it at the same time. So you're always going to have a competition which is going on between these two. And again, very well understood for these RNA phages, quite well understood for this big pink section in this textbook, or say the taxonomy book as well. Again, what's a phage? Something that makes bacteria sick that can go through a filter which blocks all bacteria. And the bacteriophage was actually coined by Félix Terrel, was a French-Canadian who moved back to Europe and really popularized the use of bacteriophage. And anyone who would like to read more about it, there's only about 200 pages in French here, which you can take a look at. So this is Félix Terrel. Twarts in England is credited with having the first discovery of these filterable things that make bacteria sick, but partly because of the First World War and potentially because he was interested in other things, didn't get anywhere near as much press as Félix Terrel, who also was quite a salesman. Must be the mustache, I think, because make that possibility. So small RNA phage. Again, we basically talked about this before, but it's really all about translation versus replication, and that gets back to RNA structure. So RNA secondary structure. Yes, it's a single molecule. I would much prefer to call these single molecules of RNA rather than single-stranded RNA, because it's all about secondary structure, so how that RNA forms double-stranded pieces. And that's all about what we're talking about in terms of how these guys replicate. This is a structure of one of these RNA phage, five-fold axis, right here, 1, 2, 3, 4, 5. Five-fold axis down here, 1, 2, 3, 4, 5. Six-fold axis in between, so we're going to have AT equals 3 particle, exactly. Two basic flavors of these single-stranded RNA phage, F2 and Q beta. These guys were found in sewage treatment plants. Wonderful place to look for bacteriophage because that's where the bacteria are. If you're going to go and look for viruses of bacteria, go and look where you have lots of bacteria. Anywhere from 100 to 10 million per milliliter. And it turns out this is a really good way to check for sewage contamination of water. And even today, when there's something leaking out on the beach, you are down at the beach on Seaside, Cannon Beach, see this flow of liquid coming out down the beach, wonder what it is. One way to check it is to look for these single-stranded RNA phage in there. Just what kind of plaques do you get on F plus E. coli? So that was, it's another way, it's a way it's still used for looking for sewage contamination. It's also really easy because these viruses don't infect people. They don't cause any disease in people, whereas a lot of the other diseases, which is why you're checking for sewage contamination, clearly can. These were just discovered originally as F-factor dependent phage of E. coli, and these guys gave nice clear plaques. I haven't really talked too much about clear plaques before, but plaque, again, your plaque assay, again, mixing together small number of virus together with a massive number of cells, go through a couple of rounds of replication. If you have a clear plaque, that means that these virus infections have led to lysis because there's nothing still growing there. We'll talk about turbid plaques a lot more later on, and turbid plaques are what happens when you haven't actually completely killed off and lysed the cells. So these are lytic phage that are dependent on the F or fertility factor in E. coli. We'll talk a little bit about Q beta a little bit later on, but the main reason that people were really interested in studying these things and why you can write a whole book about them is because they're great sources of RNA. You can get really large quantities of pure, uniform RNA that you can then study in the lab just by purifying what you found in this sewage. Again, 10 to the 7 particles per mil just straight out of your sewage treatment plant. If you grow them in the lab, you can get 10 to the 12, 10 to the 13 per mil really quite quickly. And that gives you one specific RNA, so it's really easy to work with the RNAs, and then you can reverse transcribe these into cDNAs. What do those RNAs look like? They're pretty simple. 4 open reading frames in them, and we'll talk about each of them as we move through here. Maturation protein, coat protein, replicase, and then a lysis protein. One of the things you'll notice here, because of the way this is diagrammed, this lysis protein is actually coded for in a different open reading frame than all of these other proteins right here. There's a read-through protein here which actually coded for in the same open reading frame in Q beta, but basically the process is very much the same. Maturation protein, coat protein, and replicase. So 4 proteins, and it turns out we don't even know what this A1 protein does, so it's really just 3 proteins, which seem to be important for putting this together. Again, it's a T equals 3 structure. This is the structure of, in fact, a virus-like particle. Doesn't have any genome that's associated with it. That's partly how they were able to get really nice crystals. This is actually from an X-ray crystal structure of one of these virions. It's also missing the maturation protein. If you look at virions, it turns out there's 1 copy of the maturation protein that you have in individual virions, 180 copies of the coat protein. Why 180? How do you know that immediately? T equals 3 times 60, exactly. And then 1 single-stranded RNA, which is present on the inside. This immediately tells you that there's an issue with how you're going to translate any of those individual proteins. You just need 1 maturation protein, you need 180 of the coat protein, but you just have 1 genome, which has those genes in it. So we'll see that regulation in just a second. Yeah?
Can you point out where the 6-fold axis of symmetry was again?
So 6-fold axis of symmetry can be right here. And in fact, why don't we do the pen approach here, and we can literally draw it on here. So what color do you want? Red? Purple? Will that work? Cyan. Well, sort of bluish. But there's your 6-fold right here. Of course, I can't draw hexagons on my... So we'll draw the little 6 number here. Okay, and there's our pentagon up here. 1, 2, 3, 4, 5. Yes? Good. It's now recorded on YouTube for eternity, or at least until they sell it off. So these particles, how do they interact with their host? What's the receptor? Turns out that the receptor is not surprisingly something which is specific to the F protein, because these were F factor. This is the fertility factor in E. coli. So male E. coli, female E. coli, transfer of the F plasmid, the fertility factor, that gets transferred through these pili, which are produced only in cells which are expressing the F plasmid. This normally, you can see a pilus right here that is not bound to virions. Here, at what must have been a really high MOI infection, it's completely encrusted with each of these virions. So here are all these virions. Each of these guys represents one virion. So massive amounts of virions here. Here's a nice negative stain. Remember, negative stain, you're just going to see the background, an outline of each of the individual virus particles. You're just seeing the stain, uranyl acetate in this particular case. What binds to the pili turns out it's the maturation protein. It's not the coat protein which does that. And so if you were to look at one of those virus-like particles that we just saw last time, they did the crystallography on, that's not going to bind to pili. So you have to have maturation protein. If you don't have maturation protein, it doesn't bind to pili. Once you have this binding, there's yet another proteolysis event that takes place, and that proteolysis event then allows the RNA to be brought inside the cell. Yeah? So these are all going to be enzymatic, and the enzymatic process, however, here seems to be present in the maturation protein. So it's a self-proteolysis. So you could argue whether that's really enzymatic, because once it's done, it's a single cycle. But basically, it's going to be a protein derivative. It's not going to be some kind of autolysis which takes place. And it only happens after you have interaction with the pilus. So now we have our RNA inside the cell, here, this black line. And that you have to make 180 coat proteins, one maturation protein, a few replicases, because these are really true enzymes. They go through multiple cycles. How do you do this? And you have the problem that all RNA viruses have, in that replication and translation are fighting with each other. Because what direction do all polymerases go? 5' to 3'. What direction does the ribozyme go? 5' to 3'. So these guys are going to run into each other because this polymerase is going to be moving relative to the template strand in the opposite direction, from 3' to 5', because it's always making, again, this should be the 5' end, making to the 3' end. These guys are going to run into each other. So you can't undergo replication and translation at the same time on your same genome. So how do you deal with this? And the answer is, as is true with almost all RNA viruses, secondary structure of your RNA. So again, single molecule RNA virus, I would like to say, rather than single-stranded RNA virus, because most of the genome is in some kind of secondary structure where it is, in fact, double-stranded. So translation is a quick review in bacteria. You need to have the ribosome associating with the messenger RNA. That's a complementary sequence, which is in the small ribosomal subunit, together with whatever the messenger RNA is that's going to be translated. You also need a start codon, and you need the S1 protein, which is part of the ribosome, associating with some of the sequences in a very non-specific manner, but still has to associate with usually a now truly single-stranded piece, i.e. not base-paired, part of your RNA. Of course, if you've got all of these secondary structures, you're going to have problems in terms of associating with some of these AUGs, like we have up here. Here's an AUG, there's a termination site, here's another start site. Not surprisingly, these proteins that you need a lot less of, the replicase and the lysis protein, are in these secondary structures, whereas the coat protein, remember how much of the coat protein you need? 180 copies, is not in a secondary structure. So just because of the structure of the RNA, how it's bound, you're going to start out with translating your coat protein. The process of translation will be pulling apart the double-stranded pieces that you have in your RNA. And in that process will now allow you to undergo translation of some of these genes which are otherwise not there. That's just shown here in cartoon form. Here's our ribosome going along, making the coat protein. Of course, as you have in bacteria, lots of ribosomes all lining up right behind each other, so you're making lots and lots of this coat protein. In theory, you can just have one genome which has been released. So that one genome, you're going to start making coat protein. As soon as you now start to disrupt some of this structure, now the shunting sequence, the start codon, and the S1 interaction domain for the replicase have now been made available for ribosomes to get onto it. So ribosomes get on, you're making a whole bunch of coat protein, you start making some of the replicase protein. The other protein which is encoded basically right in this same region is the lysis protein, but you don't want very much of that. And of course with lysis, you want to do that right at the very end of the replication cycle. So you want to have a very small amount of this lysis protein. How does that lysis protein actually get made? It turns out that it's not from release of an AUG and release of a nice Shine-Dalgarno sequence. This happens because ribosomes are lazy. And what happens here is that the ribosome doesn't actually completely dissociate from the RNA which is translating in all cases. In most cases it does. And so you have actually no production of the lysis protein. But in a few cases, the termination which happens here, so you have your stop codon, you have the end of translation, your ribosome release factors, which are pretending to be your tRNAs, which should be causing dissociation to take place, just cause dissociation of the peptide, and this ribosome can bounce back and forth a little bit before it completely dissociates from the RNA. And in a few cases, about 5% of the time, it will bounce back and forth along the RNA until it reaches the start codon for the lysis protein. And once it's reached the start codon for the lysis protein, then it'll start translating and translate that lysis protein. But it's only going to do this again at about 5% of the time. And how we know that this is the case, and this is described almost in excruciating detail in the textbook, that it's all about mutations. And so making different mutations in the viral genome and figuring out that you have maybe much later lysis, because all the lysis protein is about making enough of that lysis protein to make holes, or you have much more rapid lysis just by increasing or decreasing the distance that that ribosome has to go before it actually starts to make that. So that's our coat protein. The reason you get a lot of coat protein is because the start codon is completely available when the virus genome is released. You make the replicase because that translation allows the replicase protein to be made, and lysis, those lazy ribosomes that don't completely fall off. Yeah, in the back.
So, according to that picture, does the coat protein termination sequence is it also the lysis termination sequence?
No, so there's a different site of termination. Unfortunately, I don't have the genome here. The termination for the lysis protein is actually in the coding sequence of the replicase gene. But that's because it's in a different frame. So that's what's up here. Okay, so now we've talked about how to make those proteins, but we still have the problem of making more of the genome. Okay, we've got plenty of our coat protein, we've got some replicase protein, we're beginning to accumulate lysis protein, but we still need to make the positive strand genome, which is going to be what's being packaged. So first question you're always going to have is, okay, well, once you're double-stranded RNA, isn't that going to be a real major problem? Well, it turns out the secondary structure that you get in the genome that comes in the first place is so strong that you never get the two strands that are base paired to each other for any real long length of time. And that's because you're making a new RNA, but as that's being made, you're getting the secondary structures which are formed. The replicase protein will, once you've made enough of it, bind to the start codon of the coat protein gene. So now you're not going to be translating that, because you remember you also have the problem of the replicase and the ribosome potentially bumping into each other. So if you make enough of the replicase protein, that'll block translation of the coat protein. Once you block translation of the coat protein, you're not going to be making any more of the replicase protein, but at least the ribosomes will have enough time to get to the end and fall off. Once you have a free genome that you can replicate, now you can make the negative strand, because remember, you always have to be complementary. So you come in as positive strand, you've got to make negative strand in order to be able to make more positive strand. So how do you make this negative strand? Turns out that it's not just the replicase protein which is present in the genome. This is an RNA-dependent RNA polymerase. It also needs a number of host proteins, and all of those host proteins are, interestingly, translation proteins. So as well as blocking the initiation that you get for the coat protein gene, you are now also stealing some of the proteins that you'd otherwise need for translation and using those for replication of the genome. Why would you want to do that? Well, these are proteins, EFTU, EFTS, and particularly S1, which is part of the ribosome, that bind to RNA. So these are also proteins that are binding to RNA. You need to bind to RNA because that's what the viral replicase needs to do. It needs to bind to RNA as well. So it's doing really two things. These are helping the replicase bind to the genome of the virus, but they're also sequestering those proteins that would otherwise be required for translation. So you want to shut down translation and get yourself to the right place on the RNA. There's also a so-called host factor, and host factor was found, not surprisingly, by finding resistant mutants to virus infection, and then eventually identified again through awesome power of genetics. This host factor is a poly-A binding protein, and it binds to the 3' end of the virus genome. Remember, always going from 5' to 3', so you've got to start that minus strand at the 3' end of the genome. So that binds to the 3' end of the genome, gets the replicase there, and you replicate your way through the genome. Curiously enough, there's a extra guanine residue which is attached to the end of the negative strand of the genome, probably comes from EFTU, lots of question marks here, and then the replicase adds an extra A residue at the 3' end once it's done. This is how you get minus strand replication. Once you've done minus strand replication, you're actually in really good shape. As a virus, again, totally over-anthropomorphizing here, because now you don't have to fight with the ribosome when you're making the positive strand, because the ribosome can't do anything with the negative strand. There aren't any genes, there are no open reading frames that are on your negative strand. And so making positive strand copies from your negative strand is really easy. And so that process just needs the replicase protein, and because it's not competing with all of your translation machinery, you end up with a lot of it. Now, it turns out that, as we've hopefully mentioned far too many times, talked about helical structures, but also in molecular biology, ends are a real problem. There are lots of exonucleases around that love to chew in at ends. Probably the template is being protected by the interaction, probably of the EFTU protein at one end, but that's still pretty much open process. So we've talked about how you replicate the genome. We've talked about getting the coat protein, the lysis protein, the replicase protein, but there's still one that's left, the maturation protein. How do we get that? We only need one copy of the maturation protein, remember, per virion, so you don't need very much of it. It's encoded at the very 5' end of each of these genomes. How do you get it? Well, it turns out you only make maturation protein from newly made genomes. So after you've had replication that's taken place, you've made your full negative strand, now you're making positive strand again. That positive strand, as it's being made, gets translated. And how that gets translated is because there are two kinds of, you guessed it, secondary structures which you have in your RNA genome. So here is the normal secondary structure that you have. This is the very 5' end of the genome. Right here, very strong secondary structure, including the Shine-Dalgarno sequence and this sequence, which is binding normally to the S1 protein. So this you're never going to translate unless you've just made this 5' end, because again, you've copied from your negative strand of the genome. As you're making this, there's an alternative secondary structure which can form. And it's a shorter hairpin right here which is forming, just because of where your bases are. You just get up to nucleotide 10. You haven't made this full hairpin sequence. That allows this upstream sequence to bind to the S1 protein. You get binding by the ribosome. You'll get translation. So it's only after you've made new genome that that gets translated. Once you have all of these things together, your capsid protein is binding to the replicase start codon. That then is blocking any more of the replicase. Once you have a ton of coat protein around, then you have the lysis protein, which is slowly building up because of the lazy ribosome that's bouncing back and forth on the template. And eventually, that lysis protein makes a little hole in the cell, and these virions are released. So any questions about how that happens? Yes, you can go back and listen to it. But it's all about secondary structure. It's all about the secondary structure and translation versus replication, which is happening there. Last couple of minutes, I wanted to talk about a really neat application of using the replicase. And it turns out this is now mostly the Q-beta replicase rather than the F2 replicase. Everything we talked about before is with the F2 genome. The Q-beta genome is really similar. But the idea here is that there are small RNAs that you can amplify to very, very high amounts just by putting in this replicase protein. So you remember, making the positive strand, once you've made the negative strand, you don't need anything but the replicase. And so it turns out that there are smaller and smaller pieces, again, that people have found of RNA, that you can make with this Q-beta replicase. And amplifying RNA is actually really kind of hard. DNA is easy because we've got Taq polymerase and so on and so forth. Amplifying RNA is a lot harder. So the idea here is that you can amplify a specific RNA, and this is really a qualitative test. Do I have a particular RNA in my sample, or don't I have a particular RNA in the sample? The way that you do that, you put a complement to your target DNA in this RNA, which is going to be replicated, immobilize it, bind it to this sequence, and then amplify it. That's shown here in a cartoon mode. Here, your target RNA, that's this gray line which is right here, let's for giggles call it HIV RNA, that now is going to bind to a DNA oligonucleotide that's complementary to it. So binds this DNA oligonucleotide. You can now separate this RNA from all of the other RNAs that you have in the cell. So you have a sample, you bind it to that particular piece. But that's not enough. You can't detect that because often, and particularly in the case of HIV, you have latent infections. So very few cells have HIV in them. Very few of those cells are actually producing any kind of HIV RNA. So you need to amplify that. How do you amplify that? You put in this MDV RNA, either a complete one that has complementary sequence to a different part of this RNA, or in the newer versions, here we have two parts that have to be ligated together. Once you have this interaction here, now you can put in your Q-beta replicase. It'll make huge amounts of this RNA, but only if you have isolated this RNA due to binding to an oligonucleotide, for instance. So put in the Q-beta replicase. If you have your target RNA around, this will get amplified like mad. If you don't have it around, you're not going to get amplified. And it's just presence-absence kind of process for looking at this amplification. And it's just because we know about how these replicases work. And again, it's because that Replication from negative strand just needs the replicase. And that replicase makes a ton of RNA. So that's it for today. We'll see you on Friday when we talk about DNA viruses.